Hello and welcome to the 2024 American Psychiatric Association annual meeting, Psychiatric News Highlights video series. I'm Dr. Adrian Preda, the editor-in-chief of Psychiatric News. My guest today is Dr. Jeff Lichtman. Jeff, welcome.
Thank you.
You are the Jeremy R. Knowles Professor of Molecular and Cellular Biology at Harvard, where you're also the Santiago Ramón Cajal Professor of Arts and Sciences. You are a highly awarded neuroscientist, a member of the National Academy of Sciences, and starting July 1st, you will be Harvard's new Dean for Science. Congratulations. You are an experimental neurobiologist, and your research has been foundational in advancing our understanding of how experience alters the nervous system's organization in long-lasting ways. I'm seeing you today here following your lecture on Connectomics, Synapse-Level maps of the brain structure in health and disease. Now, to kick it off, could you tell me a bit about your professional journey and how did you become interested in this topic?
Sure. Thank you for inviting me to talk to you about this. I'm very actually interested in my biological history, not because I'm a narcissist, but because I'm so focused on the idea that our experiences change our brain and make us into who we are. So my exposure as a young scientist in my graduate work on scientific questions forced me to ask questions about how single nerve cells were talking to multiple target cells. And that was a difficult question to ask, and getting answers as a graduate student set me on my life's journey, basically, to look at the wiring diagrams in development and later in life, where nerve cells are being thought of as having a bunch of partners with whom they converse, and that understanding how that changes over time led to basically everything I've done, even though people say, well, you worked in electrophysiology, and then you worked in Rainbow, and now you're doing electron microscopy. To me, it's just a completely contiguous trend of questions and answers and more questions and more answers, all about the basic question of how brains are wired up at the level of individual neurons talking to other neurons.
Let's start with a big question. Why is the brain so difficult to understand?
Yeah, so the word understanding is another one of my hobbies of trying to understand what we mean by understanding. I think the brain is hard to understand if we think of understanding as a human desire for a kind of linguistic explanation of something that existed before language. That is, we want to explain, we want to turn into words something that exists without words, did not require words for its existence, and has many more streams of ideas running through it than any sentence or any book. Understanding a brain is sort of like me asking you, do you understand the city we're in right now, New York City? And if you said, what do you mean, understand New York City? And I said, just explain why every car is where it is, why every person on the street is where it is, why every building is where it is, why every doorway is where it is. You would say, that's ridiculous. There's so many things going on simultaneously. How could you possibly understand that? And I would say, if you can't understand New York City, you have no hope of understanding a brain, because it's far more complicated than New York City. So I don't think the word understanding is really the right word if we're using it in the traditional sense that I get it, I understand it, I can turn it into words. I think humans may have to settle for something else instead of understanding, which is a description, a complete description, where nothing is left out and where there are no more surprises, that you've described enough that you're not surprised by what you see. That is a form of understanding. It's not what most people want, but that may be all we can get in something as complicated as a brain.
Descriptive neuroscience versus experimental neuroscience. Can you talk about this?
Yeah, this is another complicated and somewhat fraught comparison. I think Most granting agencies prefer people to start with a hypothesis and generate experiments to test the validity of the hypothesis. But there are other kinds of science where testing hypotheses is not what the science is about. Archaeology, astronomy are areas where we just describe and we learn a great deal. And I don't see why neuroscience should be relegated to only experimental neuroscience, that it's a pejorative, a negative to say your work is descriptive. It should be a compliment. And most of my life, my work has been descriptive, but it's usually used by reviewers who are criticizing me for being descriptive. I want to be descriptive because that's the one thing I can trust, because it's not based on my ideas. It's based on what the animal or the tissue is trying to tell us. Just trying to read it rather than read our own logic systems for doing an experimental manipulation.
So you're a descriptive neuroscientist? And
unabashedly.
Very much so. And you've been working on a very complex description of the brain, which is connectomics. So can you talk a little bit about connectomics, what it means, and how is it relevant to what's happening with regards to mental disorders?
Yeah, well, I think understanding how it relates to mental disorders is the great unanswered question, I would say. But let me first say what it is. The brain is undoubtedly, all you have to do is look at it in a high-powered microscope, a gazillion connections between nerve cells that are enormously complicated in terms of the fact that nerve cells at very different places in the brain still manage to have connections with each other. So it's kind of it's very much like an internet network of human beings, say like Facebook or what used to be called Twitter, where you can have friends all over the world and you can talk to them. Brains are sort of like that. So you have this enormously complicated set of communications where each neuron is receiving lots of information from lots of other nerve cells, and each neuron is making a decision whether to send that information on to its followers, which may be in the thousands as well. So it gets thousands of inputs, sends its output to thousands of target cells, and every brain cell is doing that. They're enormously complicated. And until recently, it was out of the question to even ask, what is the structure of that set of communications? And now it's within our grasp to have a complete description of who's talking to who in a brain. If anyone thinks this is going to suddenly make everybody say, aha, now we understand, of course it won't, but it will tell us what is underlying everything that we do with our brains.
Mm-hmm. So how far advanced are we in understanding the connections?
So in terms of describing the connections, we have now tools that, at least in principle, could do something perhaps as large as a whole mouse brain. And by we, I mean a community of people. This is well beyond the reach of a single laboratory. It's like a genome project. This will require lots of people who are interested in this kind of work. There are many labs working on techniques, some related to staining the brains, some related to cutting the brains, some related to imaging the brains, some related to the computational challenges of aligning, stitching, and ultimately segmenting brains and then annotating those cells. There's just an army of different kinds of scientists necessary to make this happen, but all the parts are there for the first time, and the government has begun funding grants to allow us to at least consider doing something as big as a whole mouse brain, mm-hmm. which is enormous. That will be, 1 million terabytes. It's a ridiculously large amount of data.
So that's an enormous amount of data.
And it's an exabyte, a word that most people don't use every day.
So that's a brain mouse.
That's a mouse brain. A mouse brain.
That's a mouse brain. So human brain.
Yeah, and then you got to multiply that by another 3 orders of magnitude.
Right.
So that's a zettabyte. And that, you're now getting into a territory where the amount of data you're talking about is comparable to the kind of data that comes out of everything in a year. Everything. every company, everything, every digital thing going on, and that's just a little too much. There's also some, let's say, ethical issues about getting a well-preserved human brain. How would that exactly happen? The tissue we use is very pristine. It's preserved very quickly. So post-mortem brains at the moment are not quite as good. And there is some wondering whether maybe post-mortem brains, human brains, could be used. But at the moment, even doing a mouse would be a true milestone, I think, for neuroscience, because it would give us for the first time a sense of what we're talking about when we say neural circuits, which everybody talks about, but now we would have a complete neural circuit.
A prediction, by when do you think that's going to be?
So the government is have grants started now that are 5 years, which are the proof of principle grants, to see if any of the technical approaches that people have wondered about trying have legs. Can you do something that's not a whole brain, but big enough that one can imagine just by scaling it with more machines, you could then do a whole brain. So we're doing 10 cubic millimeters. We, again, a consortium of laboratories in a number of different universities, are doing something that is about 10 cubic millimeters of hippocampal formation. That's the hippocampus and the entorhinal cortex and some small areas in between. And if we can do that, then in 5 years, maybe the government, if they have money to do this, and all the other uncertainties related to government funding, they would then institute a project to do a whole mouse brain.
And then in 5 years, that would also require enormous computational resources and power. Are we there yet?
So, yeah, of course, this is another area that is changing very rapidly. My main colleagues on this have been Google, which are tremendously talented scientists with a unimaginably powerful infrastructure behind them. And yeah, and they're interested in seeing this all the way through. You'd have to ask them why they're interested in it, but I'm happy they're interested in it. I see, some tremendous value that would come out of this for neuroscience, but perhaps also for computer science, having an idea how brains actually work. Brains use almost no power compared to the huge requirements for power for these artificial neural network systems that are used for AI. The power costs of this is just enormous. Our brain is like a 30-watt light bulb. It's nothing, basically. And a mouse brain is even far less than that, a very dim Christmas light, basically. So, yeah, learning how brains can manage to do all this amazing compute with almost no energy is also a deeply interesting question. Maybe there's some secrets and some tricks that brains use that they would like to know.
Yeah. So your work in the human brain, where is that at this time?
So this week, a paper is being published in Science magazine that is a collaboration between Viren Jain's group at Google and ours to do a cubic millimeter of mouse brain. And that was from a part of the temporal cortex. Now we're working on frontal cortex, samples that come from patients who are undergoing deep brain stimulation for any one of a number of disorders, and usually to get the wire in through the frontal cortex, a column of the frontal cortex has to be removed or destroyed, and instead of cauterizing it and destroying it, we are taking it from a group at Mount Sinai. So that is very exciting. We have some data sets already in the pipeline from frontal cortex. And that's interesting from a psychiatric standpoint because the frontal cortex and schizophrenia have a long, fraught relationships, with lobotomies being a treatment with some controversy, I must say. But presumably there is something about the frontal cortex and schizophrenia, and there is also some interest in now deep brain stimulation as an approach for schizophrenia. So it is possible we will have eventually frontal cortex tissue from schizophrenics. And that would be interesting, wouldn't it?
Your findings so far. So you are very carefully mapping the connections between dendrites and axons, and you are actually able to trace the synapses through which the neurons connect to one another. In the mapping of the synaptic mapping, any discoveries that were surprising?
Yeah, I mean, I think for me, the greatest pleasure of doing descriptive science is that many of the things you see don't look like what you expect from a textbook. And it forces you to reevaluate many of your assumptions. So in the online data site where this Data for the Human project is located, it's a Google site that is open to the public. There's a whole page of mysterious things that don't look like anything we've ever seen before. And yeah, some of them are fascinating, and we don't know what they are. Axons that make little highly complicated sort of beehive work little areas, and then they just sort of wrap around themselves, and then they grow normally, act like a normal axon, then they do another one of these, and then they grow some more. They're very rare in our data set, but it's not a one-off. There are a number of neurons that have this unusual thing, whether this is a delay line or some kind of induction related to currents, I don't know. That's one. The one that's most interesting to me right now is that among the very, very common weak connections between nerve cells and their targets, there is a small number, but almost every target cell, as far as we can tell, get one or two inputs like this, of axons that make extraordinarily powerful connections with many synapses. Some of the target cells, they innervate. Most of the target cells are weak, meaning that there is some subset of connections in the brain where a single axon probably has enough power to drive a target cell to threshold without using temporal or spatial summation, which is the standard textbook way cortical neurons are supposed to be activated. And because we found this both on the presynaptic side of the input to a cell, and its axon's output has many weak connections, but one or two super powerful multisynaptic connections, this seems to be a pathway where you can get information to flow through a network without any summation. And maybe it explains why, when a person has learned how to drive and they're driving their car and they see a red light, and they're having a conversation or they're listening to the radio, they don't have to say, that's a red light, what do I do next? They just see the red light, their left leg goes off the gas pedal and goes onto the brake pedal as if it's a reflex, even though they couldn't have known that from birth. It had to be learned, but now it's automatic. So some fast path, post-cognitive path now is there for this very complicated driving maneuver. And maybe it's through these very powerful connections. That's what we wonder about.
You also talked about incidental versus intentional wiring. Could you discuss that?
Yeah, so, I mean, the brain is really interesting because it has this vast superabundance of incredibly weak connections between nerve cells and their target cells. And one of the interesting features of them is that the axons don't seem to be picking which particular target cell to innervate. They just run straight as a line at some arbitrary direction, and then they drop synapses more or less regularly along that path. And then among those weak connections, they'll find some, so that those I think are incidental connections. In the sense they're running in a direction, they drop synapses on whatever target cells are along that path, and then every once in a while, they find a target cell, which every time they find that target cell's dendrites, instead of dropping one synapse, they drop 5 or 6 synapses. And if that same axon bumps into the same target cell multiple times, it can end up with 50 synapses on that target cell. There's some very special relationship that seems now quite intentional, not incidental. So I use that term, intentional. The reviewers of this paper insisted that we call it purposeful as opposed to intentional, as if that somehow makes it less human-like behavior, but it seems to me about the same. But yes, so there is some question about why that occurs, but unambiguous, it does happen.
It's interesting. New brains versus old brains.
You mean baby brains versus, yeah.
Exactly, baby brains versus not so baby brains.
So, in muscle, which is not brains, and I know most people interested in brains are not interested in muscle, but for me, a synapse is a synapse, and the cells making synapses on muscle are coming from the central nervous system. So they are a good way of thinking about what might be going on in less accessible areas. Baby brains, if you look at baby muscles, you find that the wiring is much more fan in and fan out in babies. There are many more connections between nerve cells and their targets. Each target cell, each muscle fiber gets convergent information from many axons, and each axon diverges to many, many different muscle fibers. Whereas in the adult, most of that is pruned away. An axon talks to a much smaller number, 10-fold smaller number of muscle fibers, and each muscle fiber is getting information from only one axon. But it's a super powerful connection, a connection that is entirely reliable and sufficient to drive the target cell to activity, very much like those rare connections we see in the adult brain. So we think that in baby brains, the wiring diagram is not poorly formed in the sense that nothing has happened yet. It's kind of the opposite. It's like it's like a dilettante. It's trying out every possible wire, every possible connection. And then with experience, most of that gets trimmed away, and a small subset of those connections get very powerful and in muscle last for the rest of your life.
So it sounds like it's almost like a lot of potential that gets sort of narrowed, funneled over time.
I think this is, I teach college students, and, they go into college for a liberal arts education. They try a lot of different things. And the hope is that by the end of their 4 years, they have pruned away areas where they think that's not so interesting, and put more of their eggs in a smaller number of baskets. And this is very painful for young students who come to college saying how many wonderful things they can do at the same time. They can play tennis, they're on the newspaper, they're excellent students, and they have all these hobbies. And then people like me are saying you're going to have to pick something and put all your eggs in that basket. And they don't want to do this. But most successful adults become, tracked into something and don't continue to be dilettantes. So, yeah, I think that is a metaphor for what's going on in their brains as well.
Mm-hmm.
Yeah.
So let me switch gears for a bit. We talked a little bit about the fact that your work really, it's processing enormous amounts of data. And there is a possibility for AI help. You kind of mentioned that a bit. So how does that work? How did that help?
Yeah, so I think people have some concerns about artificial intelligence as being potentially dangerous and potentially problematic for the future of humans. The way we use it is to simply do things that we do, just doing it faster. So we use artificial intelligence the way it's supposed to be used. In the case of many kinds of learning, artificial intelligence is sometimes used this way, where you train a classifier to mimic the way a human would do something. And you test its ability by holding back some data humans have already analyzed and make sure it gets the same answer. And if it does it as accurately as a human, then you let it do things that humans haven't done. And that approach works fine. There are forms of artificial intelligence now where there's no human training. It just sort of infers the results. And yeah, it's scary because then you're less clear what the human's job is at all in the whole process. And maybe artificial intelligence will get there in our field of connectomics. It's already pushing in that direction. But right now, we still depend very much on human training to give the artificial intelligence network an idea of what it's supposed to do with the data.
Mm-hmm. Any limitations in terms of like when you think about training, you train it on a data set, but then each brain is different.
Yeah, I mean, there's several challenges. One is that it's a kind of dullard. You have to train it a lot. Whereas a human, you would show one or two examples and they'd be off and running. And I know that from the undergrads who've never done neuroscience before. You explain what to do, and after a few minutes, basically, they're off and doing this. With these machines, you have to give them thousands of examples before they have any idea what they're supposed to do with the data. So that's one serious, I'd say, difference between human learning and machine learning. The other thing is you train it on a particular data set. And the data set we've used for the human is a data set of a patient who had epilepsy. The part of the brain we were looking at was not the source of the epilepsy, but epileptic seizures do spread. So it is conceivable that the thing we were training on in this case was a brain that has some abnormalities that are related to epilepsy, and yet we transfer that learning when we look at other brains that don't have epilepsy. And again, we do a little bit of training to make sure it's working. If it doesn't work, it presumably means that there's something about that brain that we've been studying that's different from another brain. This is an area maybe to leverage to try to figure out what's the difference between an epileptic brain and another brain by seeing why that training doesn't work so well in another brain. But at the moment, we're not there. We're just taking advantage of any training we have to make the extremely arduous task of training a classifier easier by taking previous stuff, including mouse brain material and human brain material and even fish brain material and even fly brain material. We put it all together. By we I'm saying Google, will try to leverage as much of that information to make an informed segmentation of a new data set. But there are some issues there. We are truly in the infancy. And I think this is the hard thing, for me to, I'm personally not in my infancy, as you can tell. I'm an old man, so I would like this stuff to get very, very automatized very quickly so we can move on to bigger and bigger projects. But still, this is a very early era for connectomics.
Yeah. So significant challenges and then fantastic opportunities, I would say. 10 years from now, where do you think we're going to be?
Well, I hope 10 years from now we will be finishing, or at least in the process of completing, a whole mouse brain.
Really fantastic.
And that would be great. And I hope by then these machines will be common enough and these approaches popularized enough and simplified enough that anyone doing neuroscience on any animal, on any part of any animal, at any age, any species, will have in their background of that tissue not just the genomic information and the transcriptomic information, but also the connectomic information. It just should be part of the data set that you bring to bear before you start asking the interesting questions. At the moment, it's cutting edge, but sooner or later it should just be part of, like a doctor weighs you when you go to the doctor's office. If you have a tissue, you get the connectome before you start asking questions about the circuit.
Jeff, I want to thank you first for your tremendous work, which I know that you don't like the term, but I think it really helps our understanding of the brain. And thank you for making the time to talk to me today.
My pleasure, Adrian. It was a pleasure. Thank you.
Thank you.